Good morning, everyone. My name is Tawshik Iqbal, and today we'll be discussing effects of NOS and NOx inhibition on lunar dust simulant toxicity in human lung cells. Oh, am I hitting the right button? Yeah, the green one. Oh, it's not working. Do you mind hitting next for me, if that's okay? Thank you. So as we know, as a result of the Apollo program, astronauts came back from the moon and they had several respiratory issues and ailments. And later it was discovered that the lunar dust was the cause of this. And lunar dust was therefore deemed to be toxic to the human body because of the micromedium bombardment and ionizing radiation that occurs on the surface of the moon. It causes it to be charged, and once it enters the body, obviously you have several issues that occur. And the reason why we should look into lunar dust toxicity is because we plan on sending astronauts back to the moon through the Artemis program quite soon. So in this investigation, we use lunar dust simulants and not actual lunar dust. The lunar dust simulants mimic the properties of the lunar dust, but in particular, we're looking at lunar Highland simulant 1, also known as LHS-1. And the reason for that is because one, it was deemed to be the most cytotoxic lunar dust simulant in this investigation. And two, in previous investigation and studies, it was determined to invoke a mitochondrial ROS response or reactive oxygen species response. And so speaking of ROS, we have to talk about superoxide. Superoxide is a form of ROS that is produced by the enzyme known as NADPH oxidase or the NOx enzyme. And superoxide has the ability to become another form of ROS, also known as the hydroxy radical. And these are detrimental to the body, not good. And there is a known inhibitor used in the labs known as aposinin. And in this investigation, we used that. And so superoxide formation, ROS formation occurs when there's any sort of cellular stress. And in our investigation, the cellular stress was the lunar dust simulant LHS-1. And so now speaking of ROS, we also have to talk about nitric oxide, which is another pro-inflammatory molecule that is produced when any sort of cellular stress is produced. So nitric oxide is produced by the enzyme nitric oxide synthase or NOS, and two known inhibitors that are used in labs for the NOS enzyme is called L-NMA and L-NNA, which I'll refer to as L1 and L2 respectively for short. So yeah, those were also used in this investigation. So now, understanding this background information, we come to our question and hypothesis. So the idea is that since we have this lunar dust simulant toxicity occur, and we know that ROS is formed and NO is formed, we have to understand the idea is that can we mitigate the effects of the lunar dust simulant toxicity if we inhibit the enzymes that invoke these responses of the ROS and NO? So that's overall what we're trying to figure out here, specifically, obviously, mitochondrial ROS. So now we're going to get into the assay that was used for this investigation. So the idea here is that we use two lung epithelial cell lines. We used A549 and BEAS-2B, also known as B2B for short. And the way the assay worked is that we plated the cells to a certain cell count the night prior to the experiment. Then we did a 1-hour inhibitor pre-treatment with our desired concentration inhibitor. Then we did our 1 to 2 hour dust treatment with the LHS-1. Then we did a 90-minute Mitosox red dye incubation. And so the Mitosox red dye binds to the ROS once it's formed within the cell. That way, the fluorescence and the mitochondrial signal, which I will get into later, can be detected by the fluorescence machine. And then we trypsinized the cells and resuspended it into a buffer. And then within the buffer is where the ROS fluorescence was detected. And so now moving forward, I will only be presenting B2B data because the A549 B2B data was very similar. And as you can see with the bar graphs that you will see, essentially the overall idea is the higher the bar, the more mitochondrial ROS that was detected, and hence higher the signal, i.e. Higher the bar. So for this primary experiment, what we did was we wanted to determine which lunar dust stimulant was the most cytotoxic, and we determined LHS1 to be the most cytotoxic after doing a pre-treatment of 5 millimolar concentration of L1 inhibitor for 1 hour, and then we did a co-treatment with the same inhibitor concentration, as well as 3 milligrams per well of the following lunar dust stimulants, which was JSC-1A, LMS-1, and LHS-1. And again, LHS-1 was determined to be the most cytotoxic, hence why we continued using it. And so now we have to discuss, L1 further. So now the idea is that in the previous slide, you notice that the dust co-treatment was for 1 hour, and you notice that the signal was there was not that significant of a difference between the inhibitor treated group and the non-inhibitor treated group. There was about a twofold difference. So for that reason, we increased the co-treatment period time by 1 hour and decreased the dust dosage that we administered to see more of a significant difference, which you see here. So in this experiment, we did a pre-treatment with 2 different concentrations of L1 for 1 hour. Then we did a co-treatment with 0.5 milligrams and 2 milligrams of LHS-1. And what you notice is that even at the varying concentrations of LNMA or L1, there was a significant decrease when there was a significant decrease in the myosox-red signals that were detected. And now moving forward, we also have to discuss L2. Again, L1 and L2 are inhibitors of the NOS enzyme, which prevents nitric oxide from forming. So here L1 was used as a positive control since we know how it works. And again, what we did was we had a higher and lower concentration of L2, and we did the co-treatment with the same concentrations of 0.1 millimolar and 2 millimolar for 2 hours. And again, we see the same effect. L2 and L1 have the same effect of decreasing the mitosox-red signal quite significantly. And now moving forward, we also have to talk about the NOx inhibitor, which is apocynin. And again, we did a pre-treatment with 0.1 millimolar and 1 millimolar of apocynin. And then we did a co-treatment with 0.5 milligrams of LHS and 2 milligrams of LHS. And we did that for 2 hours. And again, we see a similar effect, not quite as of a large fold difference with using the inhibitor, but still substantial. We see that the mitosox-red signals significantly decreased with using the apocynin inhibitor. And so now, in conclusion, from everything that we've seen thus far, we conclude the following things. So NO, apologies, I forgot to mention this previously, but NO is the NO pathway, the nitric oxide pathway, is still something scientists are trying to wrap their head around because it is such a complex pathway that could be induced literally by anything. And the fact that it has both a cytoprotective effect as well as a cytotoxic effect to cells. So from this, we can understand that NO is involved in the dust-generated inflammatory response. And we understand that the NOS and NOx activity does contribute to the high mitosox-red fluorescent signals that we've seen in the graphs, and that the inhibition of NOS and NOx protects mitochondrial function. Apologies again, I also forgot to mention that NO and superoxide actually interact with one another in vivo and in vitro, where nitric oxide can bind to superoxide to form peroxynitrite, which is again another detrimental form of reactant species within the body. And overall, what nitric oxide and superoxide does is it really affects the electron transport chain. So if we can mitigate the effects of these radicals, it is super important to do so. That way, people are not negatively affected by it. So I want to give some acknowledgments. First off, I'd like to thank my advisor, Dr. Demple, over at Stony Brook, as well as my former mentor, Jamie, and Joe and Arafat, who is also a part of the pharmacology department at Stony Brook. I also like to thank Survey for having me, Washington University for having me. And I'd also like to thank Dr. Glotch for supporting my studies. And I also like to thank Michelle, who's one of the administrators of the Rise Through project over at Stony Brook, who's been helpful for me, helping me get here and so on and so forth. So thank you.